Regulation of gene expression in prokaryotes is going to be different from the regulation of gene expression in eukaryotic organisms. And the main reason for this is that the overall structure of a prokaryotic cell and where the DNA is located at is going to be different. So if we review very quickly what the overall structure of a prokaryotic cell is, recall that prokaryotic cells, they do have a plasma membrane around the outside. And then if we talk about the structure of the DNA itself is primarily a circular chromosome, one large circular chromosome right here. And that circular chromosome itself is not going to be located inside any particular compartment. Instead, it's located in what we call the nucleoid region. The significance of this, the fact that it's located in that region, which is really exposed to the cytoplasm of the cell, is that your transcription and translation very often are going to occur at the same time. And so this will eliminate some of the regulatory points as particular options when we're talking about gene expression in prokaryotes. So what we find is that the regulation of gene expression is going to be primarily at the transcriptional level. So let's just write that here. So the regulation in this case is going to be at the transcriptional level. And so that would be really regulating the promoter and the RNA polymerase, and really whether or not we actually get the transcript, the RNA produced. Here we have a look at the typical gene structure that we're looking at inside of a prokaryotic cell. For prokaryotes, they do tend to encode a lot of their genes as what we call operons. And operons contain coding sequence, so information to make several proteins. And these will be all transcribed as a single unit. And so what this means is that there will be just one promoter in front of all of this. That will be the site where the RNA polymerase is actually going to bind. And if we do transcribe this, it's all going to happen as one single piece of RNA. So this operon area that we look at right here, this all would be called the coding sequence, or what we tend to abbreviate as the CDS. Okay, so that's something that actually is unique to prokaryotic cells, the fact that we're going to kind of transcribe all of these as one piece of RNA, even though ultimately each one of these is meant to code for a different protein. And one of the reasons that prokaryotic cells are going to do this is that all of those genes that are located within this single operon, they're going to be related to each other as far as their overall gene function. So usually these particular proteins are for making or functioning in a single metabolic pathway. And so these might be all genes that are meant to help the cell make one particular sugar, or help it to break down one particular sugar. So we would usually need all of those at one time, and this way we just coordinate the synthesis of all of those enzymes or proteins at once. So if we look more closely at how the regulation of this transcription is going to occur, we do have really two important regions that precede the coding sequence that we do want to discuss. So this one's shown in green here. This is what we tend to refer to as the promoter, and the promoter is where we expect really the RNA polymerase to bind. And if the RNA polymerase binds here and is able to kind of go right down this sequence, then we would expect to get that RNA copy that we're looking for. The other sequence that we have here in blue, this is what we refer to as the operator. The operator sequence is an area where we can have repressors bind. And so when repressors bind, repressors, their job is to really repress, or in other words, they're going to block transcription. And the way that they block transcription is they're going to prevent the RNA polymerase from really getting to the coding sequence down here. And so those will be molecules that actually come and they really sit right on top of that operator sequence. And when they do sit right on top of the operator sequence, it's a roadblock. So in this case, the RNA polymerase cannot get to the really the coding sequence or the CDS. So in that case, we do prevent transcription from taking place. This is going to be something that occurs in response to signals in the environment. So if there was something that told the cell you don't have a need for these particular enzymes and proteins at this moment in time, the repressor would sit there and it would prevent the cell from just wastefully producing the RNA molecule that would then get translated into the proteins. So that's one type of regulation that we have. And in this case, when we're talking about repressor molecules acting, this is what we call negative regulation. Besides negative regulation, we can also have positive regulation. And so positive regulation is going to be something that actually increases, or in other words, this is going to enhance transcription. And in that case, we would get even more transcripts produced and ultimately even more protein produced at the very end of all of this. So in this case, we do have the RNA polymerase. Its job is really to bind to the promoter area. So this was my promoter. So the RNA polymerase is going to bind there, and if we get good binding of RNA polymerase, let's just label that this is RNA polymerase here. If we get good binding of RNA polymerase and there's nothing blocking it in this promoter region, then we would expect that it's going to be able to come down here and actually make our RNA. Well, what is going to help us in the positive regulation part of it is that we will have additional molecules that are called activators. And activators, they do enhance the transcription or make us, help us to get even more transcription. And typically these are going to help RNA polymerase bind the promoter. So if we get better binding of RNA polymerase on the promoter and we don't have a roadblock here on the operator, then overall we're just going to expect to have better transcription. So these usually bind somewhere over here, and in this case, so the yellow is representing my activator. In this case, we get better binding of RNA polymerase, and the end result is that we get more transcription, which leads to more protein at the end. When we do talk about gene regulation in prokaryotic cells, a lot of times the term inducible comes up. And inducible gene regulation usually involves a combination of the repressors and activators. And so in this case, the two of them working together will help to ensure that the cell is only producing what it actually needs, depending on the current situation inside the cell. It's just a more tightly controlled mechanism. We still have activators that are probably going to bind either the promoter or sometimes they are actually going to, we're going to have other molecules that will affect the ability of the repressors to bind. So if you have a molecule that prevents a repressor from binding to the operator, that would also enhance the transcription of that particular gene.